Space is one of the most severe environments into which humankind has ever ventured. It has helped us discover the very limits of human physiology far from the Earth's surface. And it has helped us find medical and technological solutions to some of humanity's most perplexing physiological problems. Biological processes are extremely complex. The difficulty in understanding them is only compounded when they occur inside the human body. The physiological challenges faced by astronauts in the microgravity environment aboard the ISS, loss of bone density and muscle mass, balance disorders, sleep disturbances, cardiovascular changes, and a weakened immune system can be problematic for aging people here on Earth as well. Understanding these processes in astronauts on board the ISS generates insight into how these problems can be better managed on Earth.
We look at the immune system in space to find out what regulates turning on the immune system. On Earth, the elderly have a very big problem in resisting infection. So we're looking for new ways to boost the human immune system here on Earth. And in space flight, it's much like mathematics, where you remove a variable, and when you remove a variable from an equation, you many times can get a result. And the biologic system is much the same way. We are removing gravity as a variable, and we're able to look at the immune system in an entirely new way. In fact, today we got news that we have been accepted for a paper where we were showing how the immune system is working and a new mechanism that has never been discovered before and only was possible because of the National Laboratory on the International Space Station. And with the results, we hope to help the elderly here on Earth to resist infection.
The ISS serves as a test bed for medical researchers and bioengineers by removing the element of gravity from their investigations. This allows underlying physiological processes to be revealed and better understood. One of the microscopes on board the ISS, the Light Microscopy Module, or LMM, has contributed to research in plant growth and new advanced materials. It also has improved consumer products using techniques that simply can't be done on Earth. The LMM provides investigators a unique vantage point from which they can study both the biological processes of the healthy human body and the drugs and medical technologies which can mitigate human health when something goes wrong. Using a modified commercial Leica RXA laboratory microscope, the NASA Glenn Research Center has configured the LMM to allow for real-time interaction and manipulation of experiments in space using ground-based controls. Interaction with the experiments is easy for staff, researchers, and the in-flight ISS crew. The LMM is located inside the Fluids Integrated Rack of the U.S. Destiny Laboratory Module aboard the International Space Station. The FIR rack provides the LMM with common laboratory infrastructure, including an optics bench, temperature control, power, illumination, imaging, data processing, and an ultra-low micro-G acceleration environment. The Light Microscopy Module uses the existing electronics of the Leica microscope, supplemented with unique and innovative internal and external electronics to support remote operation, enhanced automation, and superior imaging. Motors and linear actuators motorize the microscope's manual functions. Engineers at NASA Glenn Research Center and Leica equipped the LMM with 23 micromotors, which permit remote control by scientists on the ground and reduce ISS crew time requirements in space. This tends to increase the available time for research. To enable concurrent confocal imaging and wide-field imaging, two identical high-resolution monochrome cameras are provided. One is mounted to the confocal head itself, and the other is mounted on the backport of the camera headpiece. Additionally, a small surveillance camera is mounted inside the auxiliary fluids container, where sample cells are processed and contained. The LMM can image regular and specialty microscope slides, including Petri dishes and opticells, which allow oxygen transport through a membrane and provide for containment of biological samples and sample mediums. The LMM not only helps scientists better understand biological processes, it can also be used to help understand and develop innovative ways to treat disease or to maintain good health. Dr. Alessandro Grattoni of the Methodist Hospital Research Institute in Houston is using the LMM together with a team of scientists from Methodist, the University of Colorado at Boulder, and NASA Glenn Research Center in Cleveland. They're developing an implantable device that releases medicine at a steady rate. This could help people with chronic ailments live healthier, more comfortable lives. The research team hopes their drug release system will free patients suffering from diabetes, for example, from having to measure their blood sugar several times a day in order to optimally adjust their medication. A similar device may also deliver chemotherapy drugs more precisely to a tumor, thereby avoiding toxic side effects to other parts of the body. It could also deliver pain medication in a constant stream, helping to avoid over-medicating or spikes in pain. Grattoni and co-PI Mauro Ferrari are working on a device that releases medication through a nanochannel membrane and into the body at a sustained, steady rate. Grattoni says he and others need to improve their understanding of the underlying particle and fluid physics in order to design better nanochannels for a given drug.
Having a clear understanding of how these particle and molecule diffuse throughout the system, an engineered system, that will give a tremendous advantage with respect to knowledge that we have currently, and that will allow us to develop new system and improved system for, again, biomedical application on Earth.
Currently, the microscope offers simple backlighting and epifluorescent illumination. In 2016, the FIR and LMM upgrades of a confocal scanner and a frequency-doubled neodymium YAG laser and a faster digital camera will allow image slices of a sample to be assembled in 3D and analyzed in more detail. Neodymium YAG lasers typically emit light with a wavelength of 1064 nanometers in the infrared spectrum. Inside the LMM, that beam is put through a frequency doubling crystal to yield 532 nanometers green laser light, hence the term frequency doubled neodymium YAG. The LMM aids space biologists, physicists, chemists, engineers, and graduate student researchers in achieving a better understanding of colloids, biological samples, and other materials on the microscopic level. If you're planning a project that would benefit from in-flight observations of the light microscopy module, you can use the CASES program or a NASA research announcement to get on the flight manifest.
Hello, I'm Ron Sicker, the Light Microscopy Module Payload Manager. 2015 has been another great year for microscope-based research on the International Space Station. We have contributed to award-winning research in fluids, plant biology, and thermophysics. The 3D confocal end camera upgrades to LMM are scheduled to become operational in late 2016. Before these upgrades are implemented, several new areas of research will be initiated. A medical device experiment known as Microchannel Diffusion Experiment, or MDE, was sponsored by the Center for the Advancement of Science in Space, or CASIS. This experiment was initially launched on SpaceX 7 and rebuilt by Bioserve and will be conducted on SpaceX 8. 2 biophysics experiments were selected from the NASA Space Life and Physical Sciences Division's 2013 NASA research announcement. These experiments will be initiated with LMM Biophysics 1 and 2 with principal investigators Larry DeLucas and Eddie Snell. Along with ongoing colloids research with the ACE-H2 experiment, these tests will bring the total number of LMM experiments to 16. Next year, a new NASA research announcement for materials science experiments will be announced from the Space Life and Physical Sciences Division at NASA headquarters. Since 2010, LMM science has grown from thermophysics and biology technical demonstrations to also include plant biology, invertebrate biology, medical devices, colloids, materials science, and the expansion into medical and human health. The growth of interest in microscope-driven experiments on ISS is expected to evolve further with new sample modules and confocal imaging capabilities in LMM. This work, combined with possible commercial off-the-shelf systems presently in development by our integration and international partners, will take our microscope systems through the completion of the ISS mission. The coordination of this work will be the focus of next year's Microscopes on ISS 2016 workshop. The LMM work is funded by the Space, Life, and Physical Sciences Division at NASA headquarters and the ISS Program Office at Johnson Space Flight Center through codes OZ and OB. To provide full use of LMM, we will continue to collaborate with our ISS partners in CASIS. The LMM hardware is developed by a highly skilled team at Zen Technologies in Cleveland, Ohio. Thank you for attending the 2015 Microscopes on ISS workshop.